Dear friends, it's my great honor to present our recent study here. So my topic is immunopathogenesis and antibody responses elicited by SARS-2 infections. So COVID-19 is recognized as a global pandemic. So up to now, there are more than 20 million infections of the SARS-2, and which had led to the more than 700,000 of the deaths through all of the world. So as compared to previously reported coronavirus infection, such as SARS-1 and MERS, the SARS-2 caused more infections, more deaths in more countries and areas. So you also know the host immune responses play an important role in the outcome of the various infections. As for the COVID-19, most of the people developed into the asymptomatic status. Some of them progress into the mild and moderate status. Among these mild and moderate patients, many of them further developed into the severe or critical status and even to death. So, however, we don't know what is the determinant on the various outcome of the SARS-2 infections. There is also no effective vaccine and treatment. So my first question is, what type of the immune response elicited by the SARS-2 infection? So to answer this question, we first enrolled 3 mild, moderate, or mild COVID-19 patients, and also and 6 of the severe patients. Also, we had 4 health subjects as control. So we took the Bronchial lavage fluid, called here BAF. We took the BAF as samples to perform 10X and further do the single cell RNA-seq and a single cell T-cell-seq. So the UMAP figure shows that there are total of the 32 clusters according to these typical markers. So in general, the cells can be divided into the macrophage, neutrophils, dendritic cell, NK cells, T cells, B cells, plasma cells, and epithelial cells. Then we compared the proportion of each cell subsets and found that the percentage of the macrophage and the neutrophils were significantly increased in the severe patient's BAF. But the percentage of the dendritic cells and the T cells were decreased in the severe BAF as compared to that in mild patients. So next, we further comprehensive analysis of macrophage subsets. So according to these markers, and the macrophage can be further divided into the 4 groups. They are FCN1 high, group 1 macrophages, FCN1 low, SPP1 positive, group 2 macrophages, and SPP1 positive, group 3 macrophages, and FABP4 group 4 macrophages. So what's the difference between the functions of these 4 macrophage subsets? And from the table, we can show that the group 1 and the group 2 macrophages are M1-like proliferatory macrophages. And group 3 is M2 macrophages. And the group 4 macrophage is alveolar macrophages. So we divided these macrophages based on their unique expressions, not only in the typical markers, but also for the pathways and the transcriptional factors. So finally, we found the FCN1 and the SP1 macrophage was significant increase in the severe patients' BAF as compared to the health and mild subjects. By contrast, FABP4 positive macrophages was largely reduced in the severe patients as compared to the health and mild subjects. So this data shows that the pro-inflammatory macrophage was enriched in the BAF of the severe patients. So this data can be concluded two. The first, we reviewed the single cell landscape of bronchial immune cells in COVID-19 patients by single cell RNA-seq. And we also found the pro-inflammatory monocyte-derived macrophages were abundant in the BAF from severe COVID-19 patients. So next, and we further analysis the TCR clone type within T cells in COVID-19 patients. So these cells can be divided into 7 subsets according to these markers. So very interesting, we found that the clonal expanded CD8 cells show the aggregative distribution with the transcriptional homogeneities in mild patients. By contrast, in the severe patients, the clonal expanded CD8 cells show the diffusive distribution with the transcriptional heterogeneities. So, and also, and we found in the mild patients, the most of the ZNF683 positive CD8 cells are expanded. Also, these cells have a higher amplification index. And I mean here, this clonal expanded CD8 cells have big the clonal type. Okay, so but in the severe patients, there are no preferentially affiliation for the clonal expanded CD8 cells. And these cells are just manifested a connection with a connection to the proliferating T cells. So this data shows that the highly expanded CD8 cells may participate in the controlling the SARS-2 infection. So then we went to seek the property of this defined highly clonal expanded CD8 cells in mild patients. So as compared to the non-expanded CD8 cells, this clonal expanded CD8 cells show some of the significant increase in some of the tissue resident markers, such as XCL1, CLCL6, and so on. And also these clonal expanded CD8 cells show the higher score of the tissue resident called signature scores. And the total CD8 cells from the mild patients also show the higher score of the tissue resident called signature than those from the severe patients. So this data shows that the CD8 cells in a severe patient were less expanded, more proliferative, and more phenotypic heterogeneous. By contrast, there are a lot of the proportion of the CD8 cells with tissue resident and highly expanded features were present in the mild patient BAF. And finally, we also measured the several cytokines and chemokines in COVID-19 patients' BAF. So compared to the mild COVID-19 patients, and we found that the several patients had much higher levels of the inflammatory cytokines and some of the chemokines, particularly for the L6, L8, and L1. L1 beta. So in the single cell RNA-seq data, we also found the chemokine CCL3, CCL4, and CCL7, and so on were highly expressed by the lung macrophages in the severe COVID-19 patients. But we also found the CXCL9, CXCL10, and CXCL11 were much higher in both mild and severe patients as compared to the healthy subjects. We also found the CXCR16, which band CXCR6 were highly expressed only in the health subjects and mild patients as compared to the severe patients. So we also examined the corresponding chemical receptors such as CCR1 from CCR1 to the CX3CR1 on various cell subsets. And through mapping the chemical receptor interaction analysis, we found the increase the chemical such as CCL2, CCL3, and so on by produced by lung macrophages would recruit CCR1 and CCR5 positive monocytes into the lung in severe COVID-19 patients. By contrast, in the mild patients, the increased CXCL16 will include the CXCR6 positive tissue-resident T cells into the lung. So this data shows that the lung macrophages in severe COVID-19 patients may contribute to the local inflammation by including monocytes and neutrophils through CCR1 and CXCR2, while the macrophages in the mild COVID-19 patients produce more T cell attracting chemokines through CXCR3 and CXCR6. So next question is how does the host interact with the SARS-2? So you know the development of the new tools to identify the infected cells will aid to the better understanding the host virus interactions. Although there are some several methods to mark the to trace the various infected cells in vivo, but in the case of the human samples, these tools are limited, making the passenger infected cells and viral reservoir cell type hard to detect. So, here we cooperate with Professor Adi Du from the Weizmann Institute to develop and supervise the pipeline for characterization of the viral infection in single cell and data. And we called this method as a viral track. So viral track can map the single cell RNA-seq data onto a large database of known various genomes. And also viral track can provide an annotation of the infected cell type and enable the transcriptome sorting of the various infected and bystander cells. And now, the viral track contains nearly 10,000 variants. For example, here we use a viral track analysis to specially detect the influenza A, PRA viral segment in the models. We also can identify the exact location of the splicing site on segment 7. We also found the PR8 viral segment were higher in the CD45 negative cells than that in the CD45 positive cells, which is also similar to previous report. So we also test the sensitivity and specificity of the viral track analysis in more various infection models, such as the LC-MV, VSV, HSV, HIV, and HPV. So how about the COVID-19? Here we use first we use a meta cell analysis to group the buff cells into the myeloid lineage and lymphoid lineage and epicellular lineage. And each lineage was further subdivided into the smaller subsets. We also found there are some important difference in the composition of the BAF sample from mild and severe patients. So we further use the viral track to analyze the BAF samples. And we found that the SARS-2 transcription was detected in all 6 severe samples in variable amounts, ranging from the 400 transcripts to more than 50,000 transcripts. By contrast, there is no viral rates were detected in the 3 mild patients. And the coverage analysis further revealed the majority of the viral rates mapped to the 3 end of the viral segment. Then we wonder which cells are the targets of the SARS-2 infection. We observed a very strong enrichment of the viral rates in the stellate cells and the epicellular progenitor cells. And these 2 cells are no cellular targets of the virus because they express the ACE2 and the TMPRSS2. And we also found there are enrichment of the SARS-2 rates in the SPP1 positive macrophages. And this suggests that the SARS-2 potentially infected the myeloid cells or SPP1 positive macrophages has the ability to phagocytose various infected cells or particles. So we further compared the differential gene expression between the infected or bystander SPP1 positive macrophages, and we found that the infected macrophages have higher expression of the some chemokines such as CCL7, CCL8, CCL18, and ApoE. So here we conclude we developed the viral track. This is a computational framework to analyze host viral infected map, and also viral track can sort infect from bystander cells and reveals virus induced expressions. And also we found using the viral track, SARS-2 can infect epicellular cells and cellular cells and potentially alters immune landscape in severe patients. Here we provide a model here. And in the severe patients, the SARS-2 infect potentially stellate and epicellular progenitor cells, and maybe SPP1 positive macrophages. And these cells can produce a lot of the chemokines, and which further recruit the more inflammatory monocyte neutrophils and into the lung, and which further cause the lung injuries. And by contrast, after a SARS-2 infection in mild BAF, ZNF683 positive tissue resident CD8 cells was clonally expanded and maybe control the virus infection. So the third question is, could SARS-2 infection elicit human neutralizing antibodies in vivo? So it is reported that SARS-2 can use the RBD protein to bind the cellular receptor ACE2. So we guess the anti-RBD antibody may disrupt the RBD and the ACE2 interaction and thus block the SARS-2 to infect the target cells. So, and first we collected the blood samples from the 8 SARS-2 infected and convalescent subjects and tested the plasma response to the SARS-2. And we found that the plasma from the SARS-2 patients is a reactive RBD protein and spike protein and NP proteins. But these individuals show the various big difference of the plasma response to the SARS-2. And we also found plasma could specially recognize the SARS-2 RBD without crossing reacting with the SARS-1 RBD and MERS RBD. But we found some of the plasma especially recognize the spike protein from the SARS-2, SARS-1 and MERS. So this data shows that maybe the antibodies from the SARS-2 infected patients have a cross-reaction with the non-RBD in spike protein. Finally, we found that the plasma could neutralize SARS-2, but not SARS-1 and MERS. Thus we use the RBD protein to screen the SARS-2 RBD specific antibodies from the 8 donors, and a total of the 165 distinct PCR sequence were analyzed in the study. And we found that the various donors were characterized by broad diversity, and some donors showed the clonal expansion of the antibody in the repertoire. And so this data shows that the early expression and where the antibody responses are likely important in the control of the various infection. So next we selected the 13 antibodies to measure their competition for the binding to the SARS-2 RBD measured by the SPR. And we found that there are 4 antibodies, 1F11, 2F6, 1A3, and 1A4 were the most competitive antibodies. So we next studied the antibody neutralize activities against the pseudovirus and the live and live virus. And we found the neutralized activity variable considerable with the IC15 values ranging from nearly less 1 to more than 50 microgram per ml. So we further select some of the antibodies for neutralizing activity analysis against the live SARS-2. So consistent with their respective pseudovirus findings, and 1F11, 2F6, and 1A3 demonstrated the potent neutralization activities. So finally, we try to characterize the crystal structure of the 2F6, FAB, and the SARS-2 RBD complex. So, and from the structure, we found the 2F6 primarily uses their heavy chain for RBD interaction. And in addition, we also found the 2F6 attachment clash with the ACE2 here. The clash occurs between the 2F6 light chain here. And we also found the 2F6 Fab not only band the up RBD, but also band the down conformations of the RBD. So this structure findings indicated that the 2F6 can direct competent with the spike protein for the ACE2 receptors. So, and here I summary our team work here, and these are mainly focused on the immunopathogenesis and antibody responses in SARS-2. And finally, we thank our team, Dr. Mingfeng Liao, Yang Liu, and so on. And we also thank the Professor Zheng Zhang from the Fudan Universities, Ning Shaoxia from the Xiamen Universities, and Lin Qizhang from the Tsinghua Universities. We also thank the Professor Adi Du, Aidu Amit, and Banner from the Weizmann Institute and Institute Pasteur. Okay, that's all.
Okay, thank you. Thank you very much, Professor Zhang, for that fascinating presentation and for your contributions to this research. I think it's just amazing to see the level of information and understanding that has already been achieved in such a short period of time since the emergence of the virus. So if it's okay, we'll take a few questions, Professor Zhang, and I encourage others to submit any questions that they may have to the Q&A. I'm going to kick off with a question here. Obviously, a lot of focus and interest on the development of the COVID-19 vaccine. I'm interested in what you think are the major biological challenges to the development of a vaccine.
Okay. Yeah, and I think this is a very important questions, and I think at least there are two biological challenges. And first, most of the vaccine aim to the elicited B cell responses. That is aim to the neutralizing antibodies. However, more and more data show that the T cell is also important for the control of the SARS-2. And our data also show that the tissue-resident CD8 T cells is associated with the disease control. So I think a future study also should strengthen the T cell-based vaccine. And I think at least the T cell-based vaccine should be very good supplements for the B cell based vaccine. So, and I think a T cell based vaccine is very important. So second, up to now, some of the vaccine focus on the RBD protein, and some of the vaccine focus on the spike protein, and even some of the vaccine focus on the whole virus. So, and we also don't know which target is optimal for the SARS-2 vaccine. And in addition, the SARS-2 mutation is also the big challenge for the current vaccine. And so, and we only waiting for the clinical trial to show and which target is better. Okay.
Okay. Thank you. So next question here is the change in macrophages and CD8 T cells in severe patients, is it related to age or other underlying health conditions?
Yeah, and I think it is possible, but in our study, due to the limited the cases, so we did not analysis the macrophage and the CD8 T cells changed by the age and the other health or disease status. But I think a future study, we should address these questions in more cases.
Okay, thank you. So there's a few more questions here now. Is it known if patients who have recovered from SARS have a different reaction to SARS-CoV-2 infection? You know, either better or worse?
Yeah, and up to now we have had some data to show that, and in the recovered SARS-2 infected patients, and I think most of the reactions to the SARS-2 infection is better, and in our recent study published on the JAMA, we also use the convalescent plasma to treat the severe patients. At least we treat a total of the 14 patients, and there is no side effect were observed. So, and at least I think the better is more.
Okay.
Okay.
Thank you. So you were among the first, if not the first to describe co-infection along with COVID-19, along with other common respiratory pathogens, you know, within the community. I'm just interested in your views on the importance of co-infection now, you know, and perhaps parallel tests for respiratory pathogens in patients.
Okay, I beg your pardon. I beg your pardon.
Can you hear me okay?
Yes. So
okay. I'm interested in your views on the importance of co-infection among COVID patients.
Co-infections, right?
Co-infections, yes, with, for example, with other respiratory pathogens.
Oh, yeah. And in our cohort, I found the two co-infections. One is SARS-2 with HBV co-infections. And we found in the HBV positive patients, and this co-infection can favorite to clear SARS-2. But and we also found only a case, HIV, who also infected with the SARS-2. And also this data have been published on the CID, and we found this HIV and SARS-2 co-infection patients, we could not detect the SARS-2 RNA in the early phase. And in addition, we also found the antibody in the plasma was delayed increase. I remember the IgM was peaked at 6 to 7 weeks after the illness, and this period is longer than the general IgM peaks. And so, and in this HIV and SARS-2 co-infection cases, and there are two character properties. One is early SARS-2 clearance and the delayed antibody responses. So we don't know why. I guess the early clearance of the SARS-2 may be associated with anti-HIV drugs. And in the early phase of the SARS-2 infection, some of the reports have shown that the anti-HIV drug maybe have some anti-SARS-2, and at least in here. And in addition, I also think the HIV-induced HIV-induced info alpha maybe play an important role in the clearance of the SARS-2. Okay, so.
Okay.
Okay.
Thank you.
Mm-hmm.
Another question here. So, how early into the infection process can you see the difference between the immune profiles of severe versus mild cases of COVID? So in other words, can you predict how severe the case would be based on that profile?
Yeah, and I really don't know and the answers, but in recent our study and this data have not published, and in the early phase, the immune responses very important to determine the outcome of the SARS-2 infections. So if the T cell responses is very strong in the early phase, maybe the SARS-2 can be controlled. But if the T cell response is delayed or is poor, maybe the SARS-2 infection can further progress into the severe patients. So, and this is the preliminary data, and we need more cases to confirm it.